What is micropropagation? What are the different stages which are involved in the process micropropagation? If you want to know all these things, then watch the video till the end. So welcome to a new video of AVIJIT'S biology. And in this video I am going to discuss about the process micropropagation. Actually, micropropagation is a process by which we can produce a large number of plants in vitro, and these plants will be absolutely disease free. And for those plants whose germination time is too long, we can produce those plants in a short period of time by micropropagation. So the definition of micropropagation will be that micropropagation is the artificial process of producing plants through tissue culture or cell culture techniques, and it is the practice of rapidly multiplying stock plant material to produce many progeny plants using modern plant tissue culture techniques. I have already made a video on plant tissue culture. You can watch the video from here. So let's discuss about the stages or the steps involved in this process or in the micropropagation process. So in the year 1974, Murasik outlined three major stages which are involved in micropropagation. And these three stages are: first of all, stage 1 is a selection of suitable explants and their sterilization and transfer to nutrient media for establishment. Actually, it is the initiation process of a sterile culture of the explant. Actually, in this stage, first of all, we have to select the plant material which is to be propagated, and then plant tissues are removed from an intact plant in a sterile condition, and then clean stock materials that are free of viruses and fungi are important in the production for the healthiest plants. And then the collected tissues are transferred into a growth medium containing sucrose and growth regulators, and this medium is thickened with agar to create a gel which supports the explant during growth. And now the stage 2 is the proliferation or multiplication of shoots from the explant on medium. Multiplication is the taking of tissue samples produced during the first stage and increasing their number. Following the successful introduction and growth of plant tissue, the establishment stage is followed by multiplication, and through these repeated cycles, a single explant sample may be increased from 1 to hundreds and thousands of plants depending on the type of tissues grown. Multiplication can involve different methods and media. And if the plant material grown is callus tissue, it can be placed in a blender and cut into smaller pieces and recultured on the same type of culture medium to grow more callus tissue. And if the tissue is grown as small plants which are called plantlets, then hormones are often added that cause the plantlets to produce many small offshoots. And after the formation of multiple shoots, the shoots are transferred to rooting medium with a high auxin cytokinin ratio, and after the development of roots, plantlets can be used for hardening. And if there is the high cytokinin present in the growth medium, then it induces the formation of shoots, and if there is presence of high auxin in the growth medium, then it induces the production of roots. And now the next stage or the stage 3, which is the transfer of shoots to a rooting medium, followed later by planting into soil. And in this stage involves treating the plantlets or shoots produced to encourage root growth and hardening, and it is performed in vitro or in a sterile test tube environment. And hardening refers to the preparation of the plants for a natural growth environment. Until this stage, the plantlets have been grown in ideal conditions designed to encourage rapid growth. And finally, after root and shoot formation in the final stage, the plantlets are removed from the plant media and transferred to soil or potting compost for continued growth by conventional methods. This is how we can generate a whole plant from a tissue of a plant, and this plant will be same as the plant which we have selected for collection of tissue for our micropropagation process. This is the different stages which are involved in the process micropropagation. So thank you for watching the video. Please like the video and share the video, and please subscribe my channel if you have not subscribed yet, and we'll meet you in the next video. Thank you.